Right, welcome back to our chapter 2. This is based on cell and its environment. We'll take a couple of questions. I want to first apologize. This page is not the clearest, and also credit to the authors of this material, Jambre Mix. Let's go. A spirogyra cell was placed in a solution. After 1 minute, the cell has been plasmolysed. The solution X was is said to be option. Let's see. To be a very strong sugar solution. Strong sugar solution. Now, everybody, let's write down. When a dilute cell is placed in a more concentrated solution, the cell wall of the plant cell would end up, or the cell wall or the cell membrane would end up shrinking. The cell membrane would shrink, and the cytoplasm will be pulled out from the cell. This is called plasmolysis. So plasmolysis is actually when there's a collapse. So I will give example. If you put salt for water, you can't go put. You could do it like you put a cell sap. Let's use an example here. Let's use like a pumpkin leaf, ugu leaf. If you notice if you put salt inside water, serious salt inside water, enough salt inside the water, you mix them a bit, then you can put the green ugu leaf inside. After some time, you go discover say seem as if the leaf they look weaker than it would normally be. Not because they don't stay long for house. Wetin dey happen be say the salt solution inside the water is hypertonic, while the sap inside the ugu leaf is hypotonic, meaning it's not as strong as hypertonic. So what will happen? It will shrink the cell sap out of the leaf outside. So what happens is that in this case the sugar solution is hypertonic. That meaning is stronger, more concentrated than the content inside the spirogyra. So what will happen? X osmosis will happen. X osmosis is spelled EX-OSMOSIS. X osmosis will happen. And when this happens, there will be a collapse of the vacuole and a shrinking of the cytoplasm from the cell. This is what is called plasmolysis. Number 2. The movement of molecule from a region of higher concentration to a region of lower concentration is called diffusion. Don't forget. Number 3. We'll go. A fresh kola nut weighing 1 gram was cut into salt solution. After 3 hours, the kola nut weighs 1.2 gram. Meaning, say the kola nut here now, the kola nut they absorb water. Let's see. The simple experiment demonstrates that. Actually, the answer is option. Let me see. A, the salt solution, no. It's not too clear, but please manage with me. The cell sap of the kola nut is more concentrated. That's why it absorbs water. Meaning the cell sap of the kola nut is hypertonic. So, example, if you carry kola nut now, put them inside clean water, what thing will happen? The kola nut go to swell up. Similar thing you do when most times, example, I give example this, not entirely correct. If you put beans now, inside, some people before they will prepare their beans or moi they will put the beans inside water. You get, after some time you go discover that the beans don't swell up. Like if you carry 1 grain of the beans, it don't swell up past normal. So what thing that happen be say the beans they absorb the water because inside the beans is more concentrated than outside of it. I'm giving an example there. So let's write. Hypertonic solution means the solution is more concentrated than the interior of the cell. Hypotonic solution means the solution is less concentrated than the interior of the cell. Isotonic, isotonic solution means they both have same concentration. X osmosis means water moves out. X osmosis means water moves out. Endosmosis is ENDO osmosis means water moves inside the cell, just like this question now. They call it endosmosis because the kola nut get more strength than the water. It go absorb the water. If they feel them, hard. Number 4, the filament of spirogyra were put into an equal volume of sugar solution X and Y. After 5 minutes, the filament from X solution had their content rounded up, while those of Y were unaffected. The experiment shows that. Number 4, the experiment clearly shows that solution X was stronger than Y. 4 is option A. Solution X was stronger than Y. So 4 is option A. Solution X is stronger than Y. The next This chapter is not too large. 5. If an isolated living cell is left in distilled water for 2 hours, it is likely to do what? Become turgid. It go absorb water, endosmosis. 5 is option D. 6. If an isolated living cell, the same question, it will become turgid. Option D. 7. Which of the following will not allow osmosis to take place? Transparent polythene will not allow osmosis to take place. 7 is option D. 8. When a spirogyral cell is immersed in a salt solution more concentrated than its cell sap, it does what? It will lose water. More concentrated, plasmolysis will happen. It loses water and shrinkage or shrivels. 8 is option D. 9. Osmosis can be defined as, let's take down osmosis. What osmosis is? Osmosis is the movement of water across a semi-permeable membrane from a solution of lower concentration to higher concentration. 9 is option D. You see, 10. Mineral salt can be absorbed into the root by, let's see, D, no, not D. It's absorbed into the root by diffusion and active transport. 10 is option C. Please, let's write down. Diffusion is a form of passive transport because no energy is needed. 11. After 30 minutes, the level of sugar solution in the above figure will, this is actually not clear. I'd like to skip this. I wouldn't want to scratch your eye. But I think this one is weak sugar solution. This side now, weak sugar solution. This side is strong sugar solution. This place is called the semi-permeable membrane. So what will happen after some time is that the one in this place, strong one would rise here. Because this one is stronger to absorb water. 12. So 11 is option B. 12. The process by which water moves from X to Y through semi-permeable membrane is called osmosis. 13. The process by which red blood cell placed in a distilled water absorb water, burst and releases its content is called hemolysis. Now, when something absorbs water, it becomes turgid. But when it absorbs water and burst, it's called hemolysis. If you carry a red blood cell and you place it inside water, for example, it go absorb water, it go burst. 13 is option D. The major reason is because the animal cell, and in this case, the red blood cell no get cell wall that will help it hold against the water. So water will enter, it's swollen, and it will burst. Salt and water are absorbed in the roots and transported to the leaves by. 14 is option D. Let's see. Osmosis through the xylem tissue. 15. When a marine fish was taken from the ocean and put into a tank of fresh water, what will happen? Notice marine fish, meaning it is more concentrated. If you are more concentrated, you will absorb water. So 15 is option B. Let's see. 15 is option B. The body of the cell would swell and burst as a result of hypotonic fresh water. 15 is option B. 16. The diagram above, they didn't give us diagram for 16 and 17, so we'll skip. 18 is not clear. I would like to stop that there. Let's see if we could try 19 and 20. I think this ends at 20. A marine protozoan is likely to have no contractile vacuole. Contractile vacuole is the organism inside single cell organisms that help them to excrete water. You get? So 19 is isotonic seawater, option A. Because if you don't have contractile vacuole, it means that water can enter and leave your body, or your body and the water level is balanced. So if you put an organism wey not get salt inside salt water or seawater where salt dey, the salt water go absorb wetin dey inside the organism. But if the organism na fresh water, the seawater na fresh water, they go balance each other out, or the river water. So 19 is isotonic water. 20. Which of the following can cause shrinkage of living cells? Shrinkage. Hypertonic solution would always cause shrinkage due to exosmosis. Exosmosis is EX, then you write dash OSMOSIS. Exosmosis. 20 has been given to you. Exosmosis. If you've gotten to this point, well done. Remember, you are supposed to jot down the point I gave, snap and send to us on the WhatsApp group. Now, if this is your first time of coming across our videos, Please note that we run a class on the WhatsApp group where you could come on YouTube, complete the series, go on WhatsApp, submit your notes, ask questions, and you'll be given a test and you'll be corrected where necessary. Thanks for watching. I'll see you in the next video.